I think it's working. Okay. We've got metabolism or carbohydrate metabolism in this way. Damn it. Oh. I know, I'm setting the mood. Okay. We've got metabolism, carbohydrate metabolism. One of the important parts of this is there's going to be 4 parts. In these 4 parts, we're going to have glycolysis, pyruvate oxidation, and the Krebs cycle, and oxidative phosphorylation, which takes place in the ETC. The first step is glycolysis, and this is something that we do as humans, and most eukaryotes do as long as you have a metabolism. This is considered an aerobic metabolism. It's going to be a complete breakdown, not glycolysis, but this entire process of glucose, which is 6 carbons, a complete breakdown, meaning in which the final acceptor is going to be an organic electron acceptor, which is going to be oxygen. We're going to get into it, and we'll get into some theory in a little bit. So here we've got our glucose. We're starting out in the cytosol of a cell, or the cytoplasm. We're going to give it 2 ATP. Those 2 ATP are going to break down, and they're going to phosphorylize that carbon-benzene ring. And that is going to produce 2 ADP. With this breakdown and release of energy, there's going to be 2 NAD pluses waiting there to accept those electrons and make sure they don't run away and they go to the ETC, right? We're investing power right now. As soon as that's done, it's all the power we need to invest for glycolysis. And now we've got fructose 1,6-diphosphate. Not important. The next thing that's going to happen is we're going to break that molecule up. We want that energy back. It's paying us our rent. Not that much rent, because we had to give it 2 ATP. We're going to give back those 2 ADPs, and then there's going to be those 2 electrons that break this apart. They break down the ring. 2 electrons get released, get absorbed by the ATP. 2 of those phosphates that were already there get packed up. We have these 2 ADP. These 2 ADP get sent back to break down the next glucose molecule. Over here now, we've got 2 other free-floating ADP that we've earned from exerting muscles, all sorts of fun stuff. And there's 2 free-floating phosphate groups as well that are ready to bond. And they bond as this electricity breaks. Well, Eric, why don't you need an NADH? Because why don't you need an NAD to absorb that electron? Because that electron is getting slammed into this ATP because it needs energy. It's going to be an anabolic reaction and an endergonic, and build it up. This is a large molecule that stores its energy. And so now we're done with glycolysis. And our end result, we've got 4 ATP produced, 2 NADH produced, and 2 pyruvate produced. But remember, we're going to send 2 of that ATP back to work. So really, we've only gotten ourselves 2 NADH, 2 ATP, and 2 pyruvates, which are 3 carbon chains. So we haven't done any carbon breakdown yet, but this is where the fun begins. With pyruvate, there's 2 types that we work with, pyruvic acid and pyruvate. Pyruvate's made of a carboxyl group, choo-choo, or the acetyl group. The first thing to go is going to be the choo-choo group. The choo-choo group is going to disappear on us when we do pyruvate oxidation. It's losing an electron. How we're going to do this is we're going to do hydrolysis. We're going to throw water into that molecule, breaking apart the carbon bond that's connecting the choo group and the acetyl group. That acetyl group is going to go on into the Krebs cycle. The choo group, is that, am I saying, no, Coo, not choo. C-O-O. I just think about a caboose for some reason.
I think of choo-choo.
Choo-choo?
Choo-choo.
No.
Because I don't know why I like it dyslexic and reads choo.
So it's Coo.
I think it's Coo.
Coo.
Because it's a C-O-O.
C-O-O-H.
Coo.
Carboxyl group. And then we've got H2O, NADH is thrown in. That's the input. All the blue is going in. Red is coming out. Red's coming out. And remember now, we're going to do this twice because we got 2 pyruvates from that one glucose. Everything that one pyruvate goes through, the other pyruvate goes through too. So we've now done this twice. I forgot an important part of this. That's okay. So this one NADH that happens twice gets sent to the electron transport chain. But now, what happens to the COOH? Besides the H2O that we added, we now are released with CO2, waste product. We excrete that through aerobic respiration. One important thing that I did kind of skip over that I fucked up in lecture was right here. This was happening in the cytosol and cytoplasm, glycolysis. This is happening in the mitochondrial matrix. So to do this, it has had to cross, this pyruvate has now crossed 2 membranes, the mitochondrial membrane and then the mitochondrial matrix's membrane. So now we are in the innermost chamber, the lumen, of the mitochondria. Happening twice. Now we've got something really useful coming over because now we've just got this acetyl group floating around. These 2 carbons, this oxygen. What the fuck am I supposed to do with that? Don't worry. Acetyl. Coenzyme A is going to help us out. And it's going to pick up this acetyl group and take it to school. Where is school? School's up here, not down here. You go to school for higher education, so you get hired. This is the Krebs cycle, which is a series in which carbon chains are broken down and reassembled. Many of these are going to be used in different kinds of tests, affirmative, negative kinds of stuff. But the first thing that happens is we lose our school bus. The school bus doesn't go to school with you. Only the students do. School bus, back. Acetyl, boom, 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 gets broken down. It gets rung through the ringer. Ring. As this process is happening, these bonds of acetyl are being broken apart. Bonds break, release energy. NAD, come pick up the energy, take it to the ETC. There's going to be 3 NAD injected and around the Krebs cycle that are picking up these things. All right, so then we're produced 3 NADH. They're going to see this. There's going to be a little free floater, FAD. FADH2 is going to be the product of FAD. It's a little bit not as good as NAD, but we still get it. That's what we got. There's also going to be an interaction of guanine triphosphate, which is the antagonist in DNA. No, it's not. G and C, A and T. Another nucleic acid base, but it's going to get rid of its phosphate, and it's going to give it to an ATP, because there's going to be an ATP here. Where's all this ATP coming? We expend a lot of ATP. How much ATP do we expend for every gram of glucose that we get? One, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. That's 19 zeros with 4 other digits in front. It's like sextillion. Sextillion ATP molecules are produced for 1 gram of glucose. So it happens a lot. And we've got about 5 seconds of ATP in our body at any given time. Yeah, Avogadro's number. So this happens a lot. An interesting healthcare question that was asked to me by David Lechuga. He asked me, what would happen if you had something in your DNA that didn't let you make good Krebs cycle proteins? You'd probably die. Probably wouldn't be alive. But healthcare applications Shout out to you, David. So let's recount, because now we've finished production of NADH. But remember, we've done this twice. So we've gotten 6 NADH from the Krebs cycle, 2 FADH2s, 2 ATPs, and 4 carbon dioxides. Right? And then we've gotten 2 NADH from pyruvate oxidation. We've gotten 2 carbon dioxides. I know we had a hard time with this class. 4, 2, 6. 6 carbon dioxides. We had 6 carbons from glucose. They're all gone now. Brush your teeth, kids. It gets rid of the sugar. We've gotten rid of our glucose. Our glucose and our carbons. We've fully broken this thing down. All right? And here we are into oxidative phosphorylation. So now we've gotten all this energy stored in these little boxes of NADH and FAD. These are FAD and NAD, they're the electron carriers that are just moving all this stuff around. They're a dump truck or a school bus. They're an electron carrier. The electron is the H. Chemistry, chemists, whoever figured that one out, shout out. I wish we could articulate it a little differently, though. Okay. We're at the electron transport chain. This is in the place of the membrane that separates the mitochondrial matrix and the inner mitochondrial fluid. And it is a membrane, so it follows the fluid mosaic model, membrane model, which means these are protein structures, quaternary protein structures that we got from ribosomes. Shout out to David, again, Mr. Lechuga, for kind of like, hey, well, what if that didn't work? You'd be dead too, probably. You wouldn't be a human. You would be like a different kind of microbe or something. That's PG to say. And now you've got this NADH and these FADHs coming along to these protein structures. This is complex 1, complex 2, complex 3, cytochrome C. Whoa. And then you've got complex 4 and then ATP synthase. These are all protein structures within this bilayer of phospholipids that are part of the electron transport chain, linking together. So NADH is going to go to complex 1, it's going to go to complex 3, it's going to go to complex 4, and these electrons are going to get pumped. I don't know why they're called proton pumps, because they're pumping the proton of the hydrogen. Chemistry. Pumping the energy, the electron. The energy that was released when this got broken. This bond was a exergonic reaction, breaking down, or catabolic. Meow meow. Breaks down everything. We got this energy jumping along. This is an active thing. We are creating an increased level, an increased concentration gradient on this side of energy. We're going to get to that in a little bit. This is going to happen through complex 1, FADH. This is what we're talking about. Not as good. It's got to go to complex 2, and then 3, and then cytochrome C, and then complex 4. And that, you kind of, the longer the chain, if you've ever done those rope workouts, the rope's not moving on the far end. So what's happening here, kind of? The electron's just not getting there. Because you say, well, we got 2 H's. Not as good. And here you could say, well, doesn't this NADH have to do the same thing? Complex 3, cytochrome C. Complex 4, pump. Complex 3, pump. NADH into complex 4, pump. Sometimes, though, we got to pump it back in here, right? Because there's so much. Complex 4 is like, whoa. We're dropping too many electrons off. So some of these guys are going to come back in here. Excuse me. Thank you. This oxygen thing, doesn't exist as 1. It only ever exists as 2. So there's going to be two oxygens, and they're going to be hanging out, right? They're friends, right? Atomic rules and stuff. But then they see two carbons, one carbon. Whoa, forgive me. Two hydrogens pull up. That's not quite enough. Two more hydrogens pull up the oxygen and say, hey, you know what? We can go be friends some other time. And they make us some H2O, thus making oxygen the final electron acceptor of the electron transport chain. And it gives us some H2O. Whoa, didn't we use H2O earlier on in the sequence right here? And remember which side of the membrane we're on here? We're on the inside of the matrix. Where does this happen? The inside of the matrix. Hydrolyzing it, right? Recycling. It's important. Cycles. Take care of the planet, it takes care of us. Okay, so now we've got all this energy, hydrogen, electrons and energy represented as hydrogens. And now we don't have a lot of that. And nature doesn't like gradients. Nature wants to make things simple. We work really hard to get it like this. But this guy, it doesn't want to do anything, right? Because we again, what's the point of this? Producing ATP so that we can exert the energy as a life form to do what we want to do. But we don't want to have to spend too much energy to make our energy. So this gradient, the physics pertaining to these kinds of subatomic and atomic units, is going to force it that way. They don't want to. They're bouncing around. They're bouncing that way. And what's on the other side of this? It's a trap. Admiral Akbar from Star Wars. It's A New Hope, episode 4. You got ADP right there. You got some phosphate because you've been eating your bananas. And ATP synthase, ACE, it's an enzyme. So guess what? It's got an active site. On this site, it's got an ADP right there. And then it's got a phosphate site. And they're going to bond right next to each other. And this E that's going to come in there is going to act like that hammer forging it together. Jeez, this is why I need a better stool. We get ATP from ATP synthase. Nowhere else in the electron transport chain do we get it besides ATP synthase. Peace. So, a quick little recap is, in total, throughout all three of these processes found in eukaryotes with mitochondria that have oxygen present for carbohydrate metabolism, you've got 10 NADH. Roughly, for every NADH that you get about 3 ATP. So 10 times 3 is 30. FADH, we had about 2 of those because we did the Krebs cycle twice, right? And they only yield us about 2. So 2 times 2 is 4. Then we got 2 ATP from the glycolysis. And then we got 2 ATP from the Krebs cycle. So then we got 4 ATP yielded from the processes of carbohydrate breakdown, not from the ETC. Besides the ATP synthase complexes 1 through 4. That's a complete confusing idea now. Fuck. And now we're at 30 plus 4 plus 4, 38. Why'd you put a dash at 36? Because sometimes Brian Haley, my lab mate, great guy, he kind of brought up a question, well, what's stopping this hydrogen from going like that way? Besides the gradients and besides all that stuff. Well, we're saying per cycle. It's entirely possible with the fluid mosaic model. Because right now we're looking at it like this, right? It's entirely possible. There's an electron transport chain. I don't like that view. There's an electron transport chain here, but then there's one that's going here. And so now you got this hydrogen jumping over to a different sequence. So we're gonna get to that 37 on the other side because we're using it. But it's not coming from the cycles. That's where that range comes from. Or maybe your NADH kind of slacked off for the day. Who knows? molecular biologists do. Have a good day.